Associate Professor and Chief of Diabetes Center and Endocrinology Clinic at University of Illinois in Chicago. Dr. Layden is a physician scientist, so he treats the patients with type 1 and type 2 diabetes and also conducts the research on clinical and preclinical areas, including the microbiome and diabetes. I know Brian for a couple of years. We have been exchanging some of our transgenic animals, and he's really a great person to help in the several ways and the scientific studies what we have been doing together. Anyways, let's welcome Dr. Layden, who will speak today on his research on nutrients derived from gut microbiome influence metabolic response during pregnancy and beyond. Welcome, Brian.
Thank you.
Floor is yours. Thanks.
Okay, you can see my first slide.
Yes.
Okay. Yes. Please interrupt me if you have any questions. So this is actually a picture of my group in Chinatown before the pandemic started. So I think this is the last time we're all together, minus a few people. I'm going to talk a little bit of a background and then talk about some of our published data and some directions. I really did mostly preliminary new data that's coming, and Project 2 is a paper that's under review, and that is an interesting project that I find just to share with you. So type 2 diabetes is, as you know, is a major health burden in our country. And what occurs in type 2 diabetes is insulin resistance increases, in particular as we age, and all the other factors that drive type 2 diabetes, so poor diet, lack of exercise, and your genetic risk factors. And so insulin resistance increases, and at a certain point, the pancreas can't keep up in producing insulin, and then your blood sugars start to rise. And insulin resistance is normally thought of as a pathological process. In pregnancy, it's actually a normal process that goes too far in gestational diabetes or gets too unregulated. So I'm going to be bouncing back between type 2 and gestational diabetes. A lot of researchers study pregnancy and gestational diabetes as a way to look at insulin resistance in different states that kind of get a better idea. So when I was a medical student, type 2 diabetes was really thought of as a insulin resistance, insulin secretion phenomenon, that there was too much insulin resistance, not enough insulin secretion. It's been realized, and I should reference Dr. DeFranzo's many reviews and work, that many other factors are playing into type 2 diabetes. Not only is it not enough insulin secretion, but it's not enough incretins from your gut being secreted, not enough et cetera, et cetera. There's new factors. And of course, since this is the microbiome group, the microbiome is a major risk factor, and that's been shown over the last decade or more now. And that's been really shown to be causative through these fecal type transplants, where you take the fecal material from humans and put it into mice, and you can take the obese microbiome and put it into a mouse and make them fat and underweight and do the same thing. But our understanding of this process in type 2 diabetes and in insulin, the development of insulin resistance and the development of obesity is really unclear. As you guys all, I'm sure, appreciate very well, is that the gut microbiome, whatever microbiome source you study, is a highly dynamic ecosystem. So for us, the gut microbiome is influenced by diet. So if you're eating McDonald's, your microbiome is going to go in the wrong direction. If you're eating good foods, it's going to be thought to be more healthy. But then there's also the host factors. Your genetic makeup has influences your microbiome, the household you live in. So all these factors play in to mediate what your microbiome looks like and probably what your risk factors are. And then there's all this crosstalk between these pathways. And so I'm initially going to be talking about the crosstalk between the microbiome and diet and these short-chain fatty acids and the receptors that are specific to those short-chain fatty acids. But later I'm going to diverge into a new direction that I'm really excited about, and it'd be great to get your thoughts and insight. So as I mentioned, we often use gestational diabetes. We often, as a diabetes researcher, study type 2 and gestational due to the similarities and the differences to understand really the insulin resistance state. So what occurs during pregnancy and in humans is that insulin resistance increases. And so if you're treating somebody with type 2 that becomes pregnant, their insulin requirements go way up. That's a normal phenomenon, but in gestational diabetes, which is diabetes that develops during pregnancy, the insulin resistance is too great. Now, the insulin resistance occurs because there's this dynamic flux of nutrients that need to be shifted towards the baby, away from the mom at different stages. And it's thought that there's a lot of hormonal factors that are mediating these changes that are, in fact, influencing the pancreas, influencing the liver, influencing the fat to lead to these nutrient shifts. Now, a collaborator of mine published this, I guess, almost 10 years ago, showing that the microbiome, similar to in type 2 diabetes and obesity, is influenced by pregnancy. So first trimester, the microbiome is more normal. Third trimester, which is the insulin resistant phase, the microbiome becomes altered. If you put it into a mouse, become more insulin resistant, similar to what occurs in the third trimester, or what occurs in what's supposed to occur during pregnancy in a normal pregnancy. But how all this occurs is really an unknown. So when I was an endocrinology fellow at Northwestern, so my whole career has been in Chicago, just hopping around from institutions right now, we did a study to look at how pregnancy changes the pancreatic environment. And so we did this gene study, and we found that a particular receptor was altered, free fatty acid receptor 2. And this receptor is sensitive to short chain fatty acids. And right when we identified this receptor, the receptor identified that the molecule that was activating it was short chain fatty acids. So I became very interested, just starting my lab at that time, to understand why would a nutrient sensing receptor to short chain fatty acids, where the short chain fatty acids are primarily from the gut, being altered during insulin resistance during pregnancy. We also found that it was altered during obesogenic-like states, so diet-induced insulin resistance. So it has pushed my career into this path of trying to understand this relationship. And so just very briefly, so a pancreatic beta cell, what occurs is it's always sensing glucose, and glucose is driving insulin secretion through this characterized pathway. Glucose gets metabolized and it leads to insulin. Well, there's receptors, in particular G protein coupled receptors, that sit in the pancreas and these beta cells, and can be activated and enhance or inhibit insulin secretion. So a classic one is coupled to a alpha subunit, which is activated. There's a GLP-1 receptor, so glucagon like peptide 1, and it enhances insulin secretion. And it's over the last for diabetes, but there's a whole class of these GPCRs that exist in islets, and one of them is the free fatty acid receptor 2. So if you know how your receptor is coupled, in particular which G protein subunit, you can have a pretty good idea of how it's going to impact insulin secretion. Now, pancreatic beta cells have multiple other functions, but we know that the one that we discovered to be expressed in islets is coupled to G-alpha-Q and G-alpha-I, two pathways that would have opposing action on insulin secretion. Now, we've explored this, and I'm going to talk more about what we think is occurring in the pancreatic beta cell. Now, a very similar receptor, free fatty acid receptor 3, is also expressed in the pancreatic beta cell, but it's primarily coupled to G-alpha-I subunit and tends to inhibit insulin secretion. So my group over the last decade is really focused on understanding these two receptors in the beta cell, and now much more beyond that. So I'm going to kind of give a summary of where the field is at, talk a little bit about short-chain fatty acids. What are they? Not everybody thinks about them on a daily basis. And then go into what has been uncovered in the field, and really what are the big questions from my perspective. So short-chain fatty acids are primarily derived by products that are digested in the gut. It's thought that there might be other sources in the body, and I'll briefly talk about that later, but the primary source is fiber getting fermented by bacteria that helps us digest our food. And these are the three primary short-chain fatty acids. Now, acetate is the one that makes it to the highest level in circulation. But within the gut, within the lumen, within the fecal material, these short-chain fatty acids are in millimolar range. And there's receptors in the intestinal epithelial cells that are sensing them, but they also get brought into the body and circulate and are sensed by other receptors, by these receptors that I mentioned in other tissues in the body. So nutrient sensing is a way that is evolved from bacteria. So bacteria are always sensing their environment for nutrients to look for their food source. And as we become more complex organisms, the nutrient sensing mechanisms have become more complex. So not only do we have receptors for short-chain fatty acids, we have receptors for other carbohydrates, for fatty acids, that are all playing a role integrating in a variety of different tissues to maintain our nutrient balance. So like in the state of pregnancy, where the balance has to shift more away from the mom at different points to the fetus as the fetus is growing. So this is a dynamic system that likely evolved from bacteria. So I'm going to be talking about the receptors, in particular G protein coupled receptors, free fatty acid receptor 2 and 3, but there are other nutrient sensing mechanisms. So there are transcription factors that are sensitive to different nutrients, as well as ion channels, but they all sit and then signal into the cell to mediate some sort of output. And so in particular in the pancreatic beta cell, the output is insulin secretion. There are other aspects I'll hint at as well that are important, but then in other cell types, such in the gut, they're playing a role in the secretion of incretins, which go on to modulate other nutrient balance throughout the body. Oops, there we go. So going back to pregnancy, so remember that we saw that this particular receptor was upregulated in the in islets during pregnancy, which was very unusual. And so early in my lab, we explored what is the potential role of this receptor and short-chain fatty acids in pregnancy in regulating resistance and things like that. So we measured before, which is the B, and then during pregnancy, which is day 15 in mice, the short-chain fatty acid level. And what you see is that in the fecal material, acetate in particular, and propionate to a lesser degree, is increased during pregnancy. And this actually also translates to what is seen in the circulating serum, which we found was interesting. And then we had, at this time, we were using global knockout mice for FFAR2. And prior to pregnancy, we measured their ability to handle glucose, which is an indication of glucose tolerance, of overall homeostasis. Prior to pregnancy in the mice, there's no phenotype for the global knockouts in the female mice and in the male mice. But during pregnancy at this day 15, we see this impairment, which is really similar to what you see in gestational diabetes. So this is a very similar test that they give to women during pregnancy to assess if they have gestational diabetes. Obviously in the mice, we give it by an intraperitoneal injection, but you see that there's this impairment in insulin resistance when the receptor is not there. But remember, this is the global knockout. And what we see is that they're not secreting insulin to the same extent. And so in the normal wild type mouse, during pregnancy, insulin levels go high. So normally insulin levels are pretty low, in particular in female mice, but during pregnancy, the insulin levels go high because of the insulin resistance. But in these knockout mice, it did not. And so we thought, because of these data, that this was primarily due to a lack of function at the level of the beta cell. Now we've gone on further with this global knockout model to look at insulin secretion in isolated pancreatic beta cells, or really the islets, which is the cluster of beta cells and some other cells. And we've shown that acetate is mediating insulin secretion. So this is the wild type versus the knockout, and this is at low glucose, high glucose. High glucose is when insulin is secreted. Low glucose, there should be only very minimal. And then when you add acetate, we see this enhancement. And in our hands, we showed that it's actually through this particular receptor. So you see in the knockout, this enhancement has disappeared when acetate is added. So this is our model of what we observed, this relationship between the microbiome and islets during pregnancy. And I'm going to talk a little bit later about changes that occur in the microbiome during pregnancy that we're still really heavily investigating. But what you see is that the microbiome does change. There's some shift in the microbiome during pregnancy. And we see a change in the short chain fatty acid levels, in particular, increased in acetate. Now, within the islets, there's an increase in the abundance of this particular receptor, FFAR2, and with the increase, there's enhancement of insulin secretion. So for some reason, there's an increased sensing mechanism that is occurring during pregnancy, during insulin resistance, that does not occur. Now, this field, not surprisingly, as many, has a lot of opposing findings. So the global knockout mice that we've used to investigate don't have much of an obesity phenotype in our hands. So on normal diet, they don't get overweight compared to the wild type. And when they're on a different high-fat diet, we don't see a major effect. However, there is one group that sees that the knockouts get very profoundly overweight. And the other groups seem to be more similar to us, but there are other opposing findings. So the other thing that we noticed in our hands that limited our model to a great extent is that the pancreatic beta cell mass. So if you take out the pancreas, there's a certain amount of islets there. So in the global knockouts, we actually saw that even prior to any intervention. And what I mean that is pregnancy or giving them an obesogenic diet, they had a diminished beta cell mass, so that the receptor, for some reason, was playing a role in establishing the number of beta cells. Not everybody has seen that. And further, not everybody has seen that FFAR2 in particular is enhancing insulin secretion. So a couple of the groups have seen this, and there's one more prominent paper in the field that actually saw that it is actually inhibiting insulin secretion. Now, it's definitely possible because both of these receptors do have a preference, can couple to inhibitory G proteins. So let me just, sorry, a little tricky. So this other paper that I mentioned, their central kind of premise was what is occurring is that both these receptors are inhibiting insulin secretion. And they show in their study that glucose is actually being metabolized through the Krebs cycle and producing acetate, which is then signaling through these receptors. Now, we've actually haven't published these data yet in the process because it's a really an important point is acetate being made in different tissues, in particular nutrient sensing tissues. And so in their paper, they show that high glucose is actually driving acetate, which is signaling. Well, the problem with that is a couple things. It's actually known in the islet biology field that there are not any transporters for short chain fatty acids within pancreatic beta cells. Two, the way that they measured acetate was through an ELISA. And if you're in the field of short chain fatty acids, these are volatile acids. You can't actually use an ELISA. It has to be done by GC mass spec. So we actually have repeated these studies and now have gone on to do radio labeled and show that high glucose actually does not lead to increase in acetate to any substantial degree. And it's important to publish this. But the question is, why are different people seeing different effects with signaling through these receptors on insulin secretion? And so we've obviously been using tissue-specific knockouts and using other approaches to tackle that question, I'm going to show you some of our preliminary studies coming up. Now, because of those, what is the source of short chain fatty acids in the body? So fiber, as I mentioned, is the major one. Now, fiber comes in a lot of different flavors, soluble, insoluble, things like that. But so a lot of these studies early on were looking at how do you, what is the effect of an obesogenic diet in the global knockouts? Well, that's a good model, but we actually think that we also need to modulate the short chain fatty acids to really understand the pathways. So, because normally in a high fat diet, short chain fatty acids are actually pretty low. So if you look at Americans who are on a Western diet, myself included, unfortunately, I probably have very low short chain fatty acids because we don't have diets that are very rich in fiber. And so we have an international study that's going on where one of the groups is in Ghana, where they actually have a very high fiber content. They're also very protected from developing type 2 diabetes and obesity, and their levels of short chain fatty acids in their fecal material in their body is much higher. And so we think that these studies really, we need to modulate to change the level of short chain fatty acids to really understand what is the effect when we knock out these receptors. And so the last few years, what we've been trying to do to address these different findings in the field is to not only use our tissue-specific knockout, we also made a double knockout, which has its advantages and disadvantages. But we've also tried to find what is the best fiber diet and the most consistent for us to look at to understand these pathways. And so what we've done is a pretty extensive study, and this is done through Priya Dasharni in my lab, where we either gave mice a regular diet, a Western diet, or Western diet with different fibers. With fiber, the short chain fatty acid levels in the blood and the fecal material go up, but in particular in the blood, because we want to look at what happens when we alter these on a Western diet and not. So we can really see what their influence is when we activate the signaling mechanism when the receptor is there or not. And so we've done the study, and we characterize these mice quite closely or thoroughly because we want to make sure that they're doing what they're supposed to do. So it's known that fiber protects against obesity and metabolic syndrome, and so we characterize these mice. So if you want to look first at D, you see that the body weight and the control goes up a little bit. Western diet, of course, the mice get very, very heavy, but each of the fiber diets are very protective of obesity. And so likewise, they're protective against elevated glucose, which is in A, elevated insulin, which is in B. You see that all three of these here are lower than the Western diet alone. And the HOMA-IR, which is a insulin resistant indicator, is lower when there's fiber present, which is to be expected. So moving forward, we've done more studies with these fiber diets, but we've established which of the fiber we really prefer for our studies. And it's in particular, we could talk later if you want to, but it's the FOS fiber, and why we did that, I can talk about it, but that's the diet, what the Western diet with and without this fiber we're going to use over the coming years to understand these different tissue-specific knockout models that we're looking at. And so we're exploring both FFAR2 and FFAR3 through intestinal knockouts. So in the gut, 1, 2 within the pancreatic beta cell, as well as in a variety of other tissues that I'm not going to talk too extensively about, but one of them is also the adipose tissue, which does highly express, in particular, FFAR2, but we don't see much very dramatic finding with this knockout. So first of all, we wanted to see what happens when we knock out the both of these receptors. So we had to make a very specific model because you can't just cross them because both the receptors are next to each other on the same chromosome. And so you see that both receptors are knocked out in the different tissues. So the blue is the different tissues. They're not present. And this is what the weight change that we've observed so far. So right now I'm just going to give you a few snapshots of our data, because then I want to go on, switch gears a little bit to show you a new project that has emerged. And so what you see here is the wild type on Western diet, the knockout, the double knockout on Western diet. There's not much different at 4 weeks of this diet. Similarly, at week 8, a little bit more weight gain in the double knockout. But interesting, the wild type with Western diet, with the fiber, are protective, as we would expect, but the double knockout is not. So we're trying to explore why this is through different approaches, whether it's a energy expenditure, and of course we think it's multifactorial. That it's probably because we know that now we have some preliminary data that one of these receptors that is actually expressed in the central nervous system. One of them is actually in the vagal neuron, that there's not only a central nervous system, but also a peripheral tissue phenomenon that's all feeding into this the phenotype. So now, remember before when I talked about the global knockouts for FFAR2, we saw primarily impaired glucose tolerance during pregnancy. We did not see much of a difference in the male mice, whether or not they were on a regular diet or a obesogenic diet. So the left here is the regular diet. This is on a high-fat diet. We only see that they become impaired during pregnancy for some reason. Now we're in the process of putting these mice on a Western diet with fiber to see if we can more extract or pull type to a greater extent. But what you see is, so I should have mentioned that it is a true knockout. It's not completely knocked down, and that's not surprising because there are other cell types in the islet. So these are isolated islets where we looked at expression of FFAR2. But we see a very similar phenomenon in insulin secretion that we saw before. We see that there is a impairment of insulin secretion in the FFAR2 knockout. But what we're really doing now is focusing on the Western diet with fiber, this one that really modulates acetate levels in particular, and then doing in vivo insulin secretion studies where we really use the gold standard, where we're doing clamp studies to insulin secretion. Now this is the other receptor, the FFAR3. This is showing that there is good knockout within the beta cell, and we see the opposite effect of FFAR2. And this is what we found in our hands, as well as other groups, but that the receptor, when it's deleted, there's an enhancement in insulin secretion. So it's actually, if you activate it, inhibiting insulin secretion. So we're still, in a similar manner to FFAR2, exploring the Western diet plus fiber to look at what its effects are. But I'll tell you that these mice, similar to FFAR2 beta cell knockouts, on just a regular Western diet, there's no apparent phenotype, which is in many ways what we expected. So the other two things that I want to show you before I go on is, so we have an intestinal FFAR2 knockout, and in many ways we thought that this would be a more dramatic phenotype because short-chain fatty acid levels within the gut are at much higher levels. However, you see, first of all, you see that when we knock it out, the levels of FFAR2 are very low in the different regions of the gut. But when we put them on a Western diet, so the orange here is the wild type, and below is the knockout. The knockouts do not gain weight, so they're protected. So the intestinal FFAR2 deletion protects against Western diet. So we're in the process of doing the fiber. We also see a very similar phenomenon with the intestinal FFAR3 deletion, that they are protected. Now, we have focused a large effort on trying to understand, is this due to their role in secretion of incretins? Now, measuring incretins is a very challenging thing. And so what's shown here is PYY, a particular incretin from the gut. You see that it's lower in the knockouts compared to the wild type on a Western diet, and a similar when it's on a controlled diet. However, we don't put too much stock quite yet in these data because these assays, these ELISAs, are highly inaccurate. And so we're working with a incretin group from the is helping us to make sure that what we think is going on is accurate. We actually think that it might be more related to their role, FFAR2 and FFAR3, in intestinal inflammation. But I'm not going to go into great detail about that. So that's where we're at in this field of nutrient sensing with short chain fatty acids. So groups have shown that short chain fatty acids mediate weight gain. We're starting to see some effect in different tissue specific knockouts. We're trying to really distinguish through metabolic cages whether some of these are effects through energy expenditure, food intake, all which could be mediated at the level of gut or beyond. We clearly see that both the receptors have a role in insulin secretion in our hand, and we're trying to address some of the controversies that have emerged. And not that we really think that any of the data that has emerged are bad data. We think actually that in many ways, in particular for FFAR2, that there's the coupling that might be, so the beta adrenergic receptor that's in the heart is actually well recognized to have coupling switching, which has a big effect on its output at the level of the heart. So you know, it's unfortunately that one of the receptors signaling is more complicated than we would like, but that's the way it goes, I guess. So the other thing is the intestinal receptors, there's some weight gain effect. We're trying to get an idea about the incretins, but at this point, and as you guys all know, as data emerges, your ideas, your hypotheses change, and we think that actually intestinal inflammation might be going on as a major factor in the Western diet that might not be occurring in these knockouts. And we have I have a few other graduates focused on the role in the adipose and the macrophages right now. So I'm going to switch back for the next 10, 15 minutes about the gestational gut microbiome. And so we actually did this study to understand why the microbiome changes to influence short chain fatty acids during pregnancy. But what happened is it actually brought us in a new direction, which I think is pretty exciting. So we took a very broad approach. We took 3 distinct mouse lines or strands from 3 different companies. So the microbiome is very distinct. We brought the female mice in, we got large cohorts, and we bred them in isolated tissue and everything at multiple time points during pregnancy, including the heart of insulin resistance, which is between day 15 and 19. And we did metabolic changes, microbiome changes, and we did metabolomics as well. And so this is just the metabolic changes. We had CD1 57 and NIH Swiss. So they had different, they each produced different number of pups. Their body weight were obviously similar in the manner that they gain weight during pregnancy, but to different degrees. The amount of fat changes. So if you're in the obesity field, subcutaneous and visceral fat are very distinct. Subcutaneous is maybe less harmful. Visceral fat is more harmful, or more metabolically harmful, I should say. And so we see very different dynamic change in both of them throughout pregnancy in these 3 different mouse lines that we used. And on the right side here is the amount of liver fat. So liver fat is also an important metabolic readout. The greater the liver fat, the more potential hepatic insulin resistance. So in particular, you see C57 had a greater amount of liver fat than the other 2. Also see is that the glucose level change, and this is was reassuring that we did this experiment correctly. So at day 0, the levels were very similar, but then there was this increase. And so there's initially this increase that occurs in glucose in humans and in mice, but then it actually dips down during the insulin resistant phase. There's really a resetting of the glucose set point in the body. The degree of the dip was a little bit different. You see the C57 were a little bit more, maybe not impaired is the right word, but had higher levels of glucose for whatever reasons, which, you know, it's not surprising different lines have different metabolic responses. we did 16S sequencing on all of the 3 lines, and we actually saw this very similar change in diversity in mouse lines, the C57, the CD1, and NIH, that there was this increase in the diversity of the bacteria, the gut bacteria, that actually returned to some level of normal in the postpartum period. That also occurred in the other lines. But it's very interesting and probably not surprising in retrospect is that the mouse type, the strand, was the most predictive, not gestational day. So this is all the different time points that we looked at during pregnancy. So I can't remember which one is which, but this is a cluster representing one of the mouse strands. This is another one. It's another one. You see that there's no separation by day occurring, that the mouse type was the biggest factor driving. And this is when we looked at each of them by different time points. You see that day 0, they cluster in a very distinct ways. And throughout all of pregnancy, G10, G15, 19, the clusters completely separate based on whether or not it's C57, CD1, and IH. So what we are hoping is that we would be able to find some common change that's going on. In particular, as a diabetes researcher, we're focused on insulin resistance. Obviously, other points in pregnancy are very interesting as well for different reasons. But we focused on day 15, 19, the height of insulin resistance, and looked at the different microbes. And you see there are some distinct driving changes. So in the 15 to 19 day, the Bacteroides was around 36%, and the other days it was around, represented about 53%. So there's a clear shifts that were going on in the whole group as taken together. However, I should mention that when we looked for specific microbial taxa that would identify that were common across the 3 mouse lines, we didn't find any that were really characteristics or the kind of the holy grail driving insulin resistance during pregnancy. So then we also metabolomics from day 0 to the postpartum period. And you see from the heat map, there's really distinct changes from the different time points going across pregnancy. And what we found is that there are some very ones that we predicted or would have guessed and would change. So in particular, it's known that, and this is just from the C57, but we see similar changes in all the lines. The bile acid pathway gets manipulated, particular during pregnancy. Steroid hormones, cortisol levels go very high during pregnancy. But the one I want you to focus on is tryptophan metabolism. This kept on popping up as a pathway that was changing in all 3 of the mouse. Now, before I focus on the tryptophan pathway, the one thing we also did, and this is with a bioinformatics partner, is they looked at the clustering to figure out what was it, the metabolites, the microbiome, the metabolomics, that was the most similar or predictive. And so on the right is actually the gut microbiome. Overall, it was the most chaotic. It was not, there was no real pattern to it when they used this particular approach to look at the changes in this particular modeling system they use on the mono-CO3. The one that was the metabolites or the metabolic data, so weight changes, insulin, was very predictive, but even more so, metabolome really changed in very predictive manners throughout. In particular, day 15 and 19 really clustered together for some reason, which was probably not what we would have anticipated. But now going back to what we discovered, and what I'm excited about is that in each of the 3 lines, the tryptophan metabolism pathway was found to be very important. And so when you do metabolomics, it's untargeted, but in this pathway, there's a lot of different metabolites that are important metabolically that we wanted to examine. When we look closely, some of the predictive molecules, so this is from the untargeted, we found that one of the metabolites, kynurenine, was increased in 3 of the lines, in particular at day 15, and then returned to normal in the postpartum period. Tryptophan itself was not changing greatly. This is all in the blood. And so tryptophan is metabolized in what's shown over here through this pathway. It can either be metabolized to tryptamine and indole, to serotonin, or it's through IDO1 to kynurenine. So what we wanted to do is we developed a targeted approach to look at these metabolites. And so we measured kynurenine in the different tissues. And what's shown here is the plasma showing that it does truly go up relative to tryptophan. And this is actually an indication of enhanced IDO1 activity. So that's the enzyme that's converting tryptophan to kynurenine. Likewise, we saw it in the Ecomaterial in the ileum, that there was an increase. So something was driving IDO1's activity in the gut, leading to higher levels of this metabolite. We investigated, I'm just showing you a few here, but tryptamine, one of the indoles, and 5-hydroxyindole acetic acid, none of them were changing. So kynurenine is really a very specific change that we observed during pregnancy related to this metabolite. And so, as I mentioned, IDO1 is the driver of kynurenine, and its levels were going up in the gut for some reason. Now, inflammation in itself actually will drive or induce the expression of IDO1. So we did see that there was an increase in some of the inflammation markers in the gut. So now the question is, well, what is kynurenine doing? Why is it important? Is it just is it truly an important factor during pregnancy? And so we used 2 models to try to understand what kynurenine was doing. So we did a model where we pharmacologically inhibited IDO1 so that the levels did not go up. And I'm going to very briefly go over this because these data were not as clear because the inhibition had actually had an effect at baseline. So the Splenda is our control, and then you put the inhibitor in Splenda, and you see that there's actually at baseline, there's a slight impairment of glucose tolerance. It's actually reversed during pregnancy, suggesting that kynurenine or IDO1 activity changes the glucose tolerance. Now, when we looked at insulin-resistant markers, AKT in particular, in different tissues, we see that they're more, in response to insulin, more insulin responsive, so more insulin sensitive, whereas the Splenda is not. So, so during pregnancy, there's some factor changing through this through this enzyme, insulin resistance. Now, we did validate that the kynurenine levels were not going up. So this is the control. You see that during pregnancy, the levels go up. It goes up slightly in the condition where we have the inhibitor, but it is statistically different. And then when we looked at it in the ileum, you see that the levels don't go up at all. And there's also less overall inflammation when we looked at inflammation in the blood. But this was really not the best model. So what we did next is we obtained a genetic knockout. It's a global knockout of IDO1, and looked at glucose tolerance prior to pregnancy, and there was no difference. And then during pregnancy, the knockout was protected from developing impaired glucose tolerance. So something was going on. So either knocking out IDO1 or having less kynurenine was leading to less impairment. And likewise, the bottom here is an insulin tolerance test, which is an indication of insulin resistance. And so the insulin resistance was higher in the wild type compared to the knockout. So there's less insulin resistance, which goes along with what's seen here. And in the IDO1 knockout, you see that the plasma kynurenine to tryptophan levels were lower at day 15 than the wild type. Even more so in the ileum, in the fecal material there, were much more suppressed when we looked at those levels. So the last thing we did, and we're still working on this as this paper is under review, is to see if we can transfer this phenotype. So we first did mouse to mouse ecomaterial transfer, and then we did human to mouse. So mouse to mouse, we took fecal material from pregnancy, then during pregnancy, and you see that in the ileum, the tryptophan, or the kynurenine to tryptophan ratio does go up. It's actually not statistically different. And when we did this transfer, there's also a greater degree of insulin resistance. So that would go along with the idea that we think kynurenine levels, the higher they are, the more insulin resistance is occurring. But we're still trying to tease out in greater detail because these data are not overall very convincing. So we next did it with human, where we took trimester 1, which is a normal, very little insulin resistance, to trimester 3, which is a much more insulin resistant phase, and did a fecal material transfer into the mice. And you see that the kynurenine to tryptophan levels were elevated compared to the trimester 1 in the control. And we do see a change when we did the insulin tolerance test, that the trimester 3 was more insulin resistant. However, like I mentioned before, we're still working on these studies to do them. So these were all done with antibiotic-treated mice first. So we're trying to do some now with germ-free mice, so we avoid some of the, I guess, side effects or complications of using antibiotics to wipe out the microbiome before we did the fecal transplants. Overall, these are very tricky experiments in my opinion, to get reproducible findings. But what we also did is with a collaborator, we wanted to see, so if kynurenine is really involved in the development of insulin resistance and is a factor in gestational diabetes, the levels should be higher in gestational diabetes compared to non-gestational diabetes. So we were fortunate we had a collaborator that had some blood samples. And we matched them by age. Obviously, the fasting glucose was different because one has gestational diabetes, the other does not. And you see that we had about a fair amount. And you see the kynurenine levels were higher in those with gestational diabetes compared to those without. So going along with what we believe would be the role of kynurenine in this process. So this is really kind of what we think is occurring, that we're working on and trying to get funding and trying to publish this first paper, is that during pregnancy, we know this from our other studies, that there's some level of gut inflammation occurring. And what that factor is, why that is a big question, and how to, I'm not sure. And I'm happy to share any of the data if anybody wants to help us explore it. But we know that IDO1's expression gets induced due to this inflammation during pregnancy. We don't know if this occurs in humans. Obviously, that's a harder thing to verify. We don't know if that there is an induction of IDO1 in the epithelial cells, but this leads to greater levels of kynurenine. Kynurenine is known to go and affect a lot of different tissues. So there's different receptors, nuclear and membrane in the liver and in other tissues that are sensitive to kynurenine, and clearly could be altering insulin resistance in the liver, in the pancreas, and in fat. So we think that these increased levels of kynurenine, so we think the microbiome is driving these changes, driving the IDO1 expression, which is leading to greater kynurenine. And then downstream, it's affecting some of these other. So one of the potential targets of kynurenine is the aryl hydrocarbon receptor, which is known to have a role in inflammation and in metabolism. So this is kind of a generic of where we're going right now. So big question is, one is how is that inflammation driving changes in IDO1? There are some people that do look at that relationship, and there's some findings. But then also, how is kynurenine leading to changes in insulin sensitivity? What are the targets? And it's probably multiple targets. And so we're doing some exploration of that to try to get an idea. And then, as I mentioned, is there key bacteria that are leading to this inflammation, or are there ones that are specifically more targeted towards IDO1? I don't think so, but that's kind of the general direction that we're going with this project. So before I end, I just want to mention the bioinformatics was Yang Dai at UIC. Jack Gilbert's been a long-term collaborator that was at University of Chicago, now is at UC San Diego, and his postdoc, who's an independent researcher now anew. We did all of our metabolomics and short-chain fatty acids through our UIC core, and I have funding through a variety of agencies, but so did this work. Maida was not in this picture. Priya Desharnais, she did a lot of the work for both of the projects. And then Caillou has done some, as well as Joseph Zapater. So really it's been a collaboration, as well as Barton Wicksteed, that has helped with our pancreatic biology studies. So it's been really a collective study or work from over the last multiple years. So I'm happy to, let me, oops, go back. Just happy to take any questions.
So as a student studying at UNF, I actually am in the dietetics program there, and I'm very interested in your findings right now currently. Some of the understanding that I have with gut microbiome is the fetal programming factor. I was curious if you've considered this in your studies and if this makes any significant findings, or perhaps a different path that you may have not considered.
Yeah, well, that's it's a complicated question for both projects, actually.
And just to add to that, what type of fiber are you guys using in your studies currently?
Okay, so the fiber that we found to be best was the FOS, which is the fructooligosaccharide. And I'm happy to share with you where we got it from. This is for our mouse studies. Now, the embryonic factor, so there was actually a very good paper looking at the role of short-chain fatty acids in these receptors and the development in the embryo of the pancreas that came out a few years ago. So, and that actually supports some of our findings that we found that those short-chain fatty acid receptors are actually important in the establishment of beta cell mass. And the establishment of your beta cell mass seems like it wouldn't be an important thing, but it's a very important thing when you're living in a Western world and you're being challenged by a Western diet. The more beta cells, in my opinion, as a diabetes physician, the better to kind of survive. So there's definitely an embryonic development going on with programming crosstalk between the microbiome and the pancreas that is very fascinating. That's, I think, just emerging. So I think that's a great field.
Thank you.
Fascinating work. Just a quick question I have is there any way we can differentiate the acetate coming from the microbiome versus the host?
Yeah, there have been people that have done some labeled studies, you know, and I've kind of always wondered if that's something that we should do to address that. And I'll also say that we have not done it yet, and that's probably just more to manpower and resources. But on that point, you know, even going further, now that we've discovered the expression of these receptors in certain parts of the brain, and is the short chain fatty acids that are circulating in the CSF, are they from the gut microbiome? I think that will be an important question to answer. But we've been doing these really cool studies that I did not show, is where we actually infuse either acutely or chronically different short chain fatty acids into the CSF. And one of my junior faculty, Ping-Wen Yu, is just amazing at doing these. I don't know how he does these, can put these cannulas in them, but it really affects appetite and other activity levels of these of the mice. So I do think at some point trying to distinguish where all these things are coming from is going to be important for short chain fatty acids. I think there's a question from somebody. Let me see if I can.
Subra, yeah.
Oh.
Oh. Hi, Brian.
Hi.
Nice seeing you again at USF.
I know, I was going to see. We've met before.
Yeah. So my question is that, as you know, diet influences immune status of everybody. So, and you know that there are particularly black 6 mice are prototypical Th1 type mice. So when you were looking at it, it was intriguing to see that there are a lot of microbiome changes among strains. So does it anyway correlate with their Th1, Th2 status?
I don't know. That's a great question. How would we? I guess we'd have to isolate those.
Yeah, you can look at their take their spleen and look at their cytokine secretion profiles that can distinguish whether they're producing a lot of IL-4, IL-13 versus interferon gamma and type of. I mean, that's Th2 versus Th1 type. Typically, that's what I mean. That would be interesting to correlate, and then you may be able to associate certain or certain fatty acids, maybe, or bacteria to this immune status, modulating immune status.
Yeah, no, that's a great idea, and I'm sure we still have the spleens, because we in general don't do much with them, but we always isolate them. So thank you.
Great, very fascinating work. So this kynurenine, which is coming out from mostly from the diet, right?
Yeah, so it's coming from tryptophan. There's no. It has to be generated through tryptophan metabolism.
Okay.
And is my understanding.
And does it have receptor or the transporter?
For kynurenine?
Mm-hmm. On the epithelial cells, yeah.
So, well, there's nuclear receptors that are sensitive to kynurenine. There's a protein coupled receptor that is sensed by that senses kynurenine. But kynurenine, and a reviewer picked up on this and kind of hung his hat on it, is that it can be metabolized to a lot of other things as well, and it clearly does. And we can't measure all of them right now. But yeah, it's affecting a lot of different tissues. And the one thing I didn't mention is there is a great work from a guy at UCSF, not a long time ago, but looking at tryptophan and the production of serotonin in the pancreas. And that actually in itself is mediating metabolism during pregnancy. We don't actually, kynurenine, we don't see a clear role in pancreas, but we think it's more liver and fat, more of an insulin resistant, not an insulin secretion phenomenon. But yeah, so we're still trying to figure out where it started.
Very interesting, yeah. Any more questions?
Well, thank you, everybody. Appreciate it.
I
Nice
just
to see
have one more question about NAD's role, NAD plus's role in, have you guys experimented in that direction as far as metabolite in cell signaling. I was really curious as to what your thoughts were on that.
No, that's a great idea. And yeah, there's definitely a role of NAD, and it's a complicated field. Definitely don't fully know, but we're kind of touching on it a little bit with tryptophan metabolism.
Mm-hmm. Thank you.
Great. Thanks a lot, Brian. It was really wonderful, and thanks everyone for attending today's talk.
Yeah, if you have anybody has any questions or needs any material information, please email me, and stay safe in Florida.
Okay, no, thanks.
Thanks, guys.
Bye.
Thank you.